So yeah, I'm Ben Tickner and I work with Simon Dockett at the University of York. And I'm going to be talking to you today about some applications of SABRE hyperpolarized NMR. This is mainly going to be in the area of hyperpolarized reaction monitoring and using SABRE to detect low concentration reaction intermediates. And the molecule that we use to do that in this case is hyperpolarized pyruvate. So I'm going to spend a little bit of time before I do that just talking to you about how we can get SABRE hyperpolarized pyruvate. So we have quite a focused audience here, so I don't need to go into any great length explaining to you why we need to use hyperpolarization, what parahydrogen is and what SABRE is. But I just really want to say or remind you that SABRE uses active polarization transfer catalysts like the ones shown on this slide here, where you break the symmetry of your parahydrogen by a reversible oxidative addition reaction. And then what you can do at low magnetic field is you can transfer polarization from these parahydrogen-derived hydride ligands to ligated substrates here. And then dissociation of these ligands builds you up a pool of hyperpolarized ligand free in solution. If we look at some of the types of molecules that are commonly used in SABRE hyperpolarization, they usually all contain N-donor sites. So it's things like pyridine, imidazoles, diazarines, imines, nitriles, and amines. So it's usually a restriction of SABRE that your molecule has to be able to bind and ligate to your iridium catalyst. But what we are interested in doing at the University of York is looking at new ways in which we can hyperpolarize molecules using SABRE. So there are two main ways in which we're able to do this. The first is using sulfoxide-based polarization transfer catalysts to hyperpolarize O-donor molecules like pyruvate. And this is what I'll be talking to you about today. And if I have a little time at the end, I'll mention briefly some relayed proton exchange effects that we can use to hyperpolarize molecules that don't even ligate to our iridium catalyst. And then once we have these hyperpolarized signals, we can use them for a range of applications. And as I've already mentioned, the one I'll be focusing on today is reaction monitoring. So I'll just take a few slides to explain how it is that we're able to hyperpolarize pyruvate before I move on to some applications. Pyruvate is a molecule that people are very interested in because you can use it for in vivo cancer imaging. You can track its metabolism in vivo. But to hyperpolarize pyruvate using SABRE, we have to overcome the weak binding of O-donor substrates to the iridium catalyst. So we have to kind of try and find a way in which we can make an active magnetization transfer catalyst that has ligated pyruvate. So our idea was that if you have any other sort of N-donor ligands present in solution, they're likely to outcompete pyruvate binding. But actually, in fact, the binding of pyruvate to a wide range of different metals has been reported in the literature. So why can't we do that with iridium? Can we use some kind of co-ligand that binds to either sulfur or oxygen to sort of facilitate or encourage pyruvate binding? So we can do that if we use a sulfoxide-based co-ligand, and this is just an example of what happens if we take an iridium-based SABRE precatalyst, doubly carbon-13 labeled sodium pyruvate, and in this example, dimethyl sulfoxide. If we add them with parahydrogen in methanol and shake them at 1 microtesla, so under SABRE sheath conditions, you can see in our single scan hyperpolarized carbon-13 NMR spectrum, you can see hyperpolarized signals for free pyruvate, shown by the blue shapes here. And in this case, they are enhanced by about 1,000-fold. But interestingly, we see two additional forms of sort of pyruvate signals here. One shown by the black shapes, and a second one, if you zoom in, you can see shown by the red shapes. So this is telling us this is probably some kind of pyruvate in two different forms that's bound to our catalyst. But the real question that we have is, what is the structure of the active polarization transfer catalyst that is giving rise to these hyperpolarized pyruvate signals? So we can get a lot more information if we look at the proton or hydride region of some hyperpolarized NMR spectrum. You can see that there is one clear complex that is giving us strongly hyperpolarized hydride signals. And if we cool these solutions down and do low temperature 2D NMR characterization, we can show that this is in fact the structure of our active polarization transfer catalyst. So you have a sulfoxide bound through sulfur. Your pyruvate is ligating to your iridium through 2 oxygen centers, and in this case, it is ligating to your iridium in the same plane as your hydride ligands. But you'll actually note that there are several also, other sort of low concentration species also present in these mixtures. So we can characterize what some of these are as well. One of them we can see is a less active isomer of our polarization transfer catalyst. So it's essentially the same. The only difference is now your pyruvate is coordinating in the plane perpendicular to your hydrides. Your sulfoxide is now trans to hydride, whereas here your sulfoxide is trans to your carbene. We also see signals shown by the green triangles for this species, which is the bis DMSO chloride species, which I'll talk more about on the next slide because it turns out to be quite important. So I want on this slide just to mention quite briefly sort of the mechanism of how this works. And when I say mechanism, I mean more from an inorganic chemist's standpoint. I've already said to you that this is the catalyst that is responsible for our pyruvate hyperpolarization. But actually, when we examine its ligand exchange processes by exchange spectroscopy, we note that pyruvate exchange and hydrogen exchange within this catalyst is actually quite slow on the NMR timescale. But actually, it's this bis DMSO chloride complex, which is also present in these mixtures, that we observe having very rapid rates of hydrogen exchange. So we expect that species of this form are very important in mediating para-hydrogen exchange within these systems. So this is quite different to SABRE with sort of pyridine type substrates in which substrate exchange and para-hydrogen exchange is happening in the same catalyst. In this case, we think that pyruvate polarization and your hydrogen exchange are happening in 2 separate species. And this makes optimization of this process much more complex, and you also have to bear in mind that your active catalyst can exist as a much less active isomer. But I just want to summarize a few sort of take-home messages from our optimization studies. We have changed, in this case, the sulfoxide, the carbene concentrations, temperature, pressure to try and increase the SABRE signal gains we can get for pyruvate. And a few points I just want to share with you. The first is that generally we find that sterically large carbenes are required for you to form this active isomer here. And then you can use the electronic effects of your carbene to sort of fine-tune the ligand exchange processes. The hyperpolarized pyruvate signal that you get is linked very closely to the concentration of this active catalyst that you have present in solution, but polarization transfer efficiency is also important. So what I mean by that is you can use 2 different sets of conditions with different carbenes and different sulfoxides, and you can have 2 different systems that generate similar concentrations of this active catalyst, but you can end up with very different signal enhancements for your pyruvate. So it's telling you it's very important to form as much of this catalyst as you can, but it's also important that that catalyst is efficient at SABRE. And the last point I want to mention on this slide is that in all cases, you eventually have catalyst deactivation to form dimers. And there's an example of that here, iridium-iridium dimers. In this case, they're bridged by a hydride ligand. But in other examples, we've noticed them bridged by the sulfoxide ligand. And you can change the time scale of this deactivation process by changing your carbene ligand and your sulfoxide ligand. In some cases, we observe deactivation in just a matter of minutes, and you no longer see any pyruvate signals. In our optimum systems, the system performs well for several hours before you get sort of significant buildup of these dimers. So I just want to sort of summarize our optimization steps more visually. So if you look on the left-hand side, these are single scan carbon-13 SABRE measurements. The signal from the site shown by the squares on the left-hand side, the material free in solution is shown here, and that site when bound to the catalyst is here. And it's a similar thing on the right-hand side for the other site. The site's shown by the circle. The free site appears here, and that site bound to the catalyst is here. So when we start out by using phosphine-based catalysts, so the conditions that are given here, we have very low pyruvate polarization levels. But if we move to N-heterocyclic carbene-based catalysts, we have quite a large increase in our SABRE efficiency. And we can increase that further by increasing the shaking time, playing around with the particular sulfoxide that we use and its concentration. So this is the optimized, sort of the optimal signals that we can get at the moment. So they're just over 2000-fold, which comes out as about 1.7% polarization. And this is in methanol. I should tell you that if we move to sort of 70-30 water ethanol mixtures, our signal enhancements decrease by around an order of magnitude. And that's going to be an obstacle we're going to have to overcome for biomedical translation of this technique. But the application that I want to spend more time talking to you about today is using SABRE hyperpolarized pyruvate to do some reaction monitoring at high field. So pyruvate has quite a diverse chemical reactivity. It's known to be converted into a wide range of different products metabolically. It can react with amines. In this example, we are reacting pyruvate with hydrogen peroxide. You form this short-lived intermediate here, which decarboxylates. You break this carbon-carbon bond and you form ethanoic acid and carbon dioxide. So our idea was, can we use SABRE hyperpolarized pyruvate to sort of monitor this reaction? Well, yes, we can. So the way that we're doing these experiments is we're taking our iridium pre-catalyst, we're taking our sulfoxide and our pyruvate, doubly carbon-13 labeled pyruvate, and shaking them for 30 seconds in methanol with parahydrogen in a mu metal shield. And then we take the lid off and inject in hydrogen peroxide. And then we put it into the spectrometer at 9.4 Tesla. And here we're just recording a series of single scan carbon-13 NMR spectra with 10-degree flip angles, which are spaced at 5-second time intervals. So you can see in these measurements, we're clearly able to detect the products of chemical change. You can see hyperpolarized peaks for the carbon dioxide, carbonate, and ethanoic acid products. The real question we have here is, can we use these measurements to extract a quantitative kinetic rate constant? Because that would be really interesting. So in order to do this, we need to do some kinetic fitting, and this kinetic fitting needs to take into account three factors that cause your signal intensities to change over time. The first is radio frequency excitation, the second is relaxation, and the third one is this peroxide reaction. So we can use these first two equations to account for radio frequency pulsing. So the signal that is available for sort of visualization using the pulse is given by this first equation, where this is your flip angle here, and the amount of magnetization that you have left for further detection is given by this second equation. In the time intervals, sort of those 5-second time intervals between your radio frequency excitations, your magnetization is changing due to relaxation here and also reaction. So we use these equations to account for both of those factors. So what happens now when we take these kinetic equations and fit them to the signal intensities from those spectra I showed on the last slide? So even though we're using doubly carbon-13 labeled pyruvate, let's just look at what happens for this labeled site first, which is converted into carbon dioxide. So we normalize it to 1 in the first instance and fit it to the kinetic equations I just showed. And you can see we get a reasonable fit. We have a rate constant for this reaction of 0.056 decimeter cubed per mole per second. And the relaxation times the model gives us compare well with some previously reported values. In this case, this carbon-13 site has a longer T1 time here, 47 seconds. So you're able to monitor this reaction out to a time window of sort of 80-90 seconds. If we now look at the same sort of signal, but sort of on the other site, the site that's converted into ethanoic acid, we see now our T1 is much shorter. And as a consequence of that, we're only able to monitor this reaction out to sort of 40 or so seconds. So because we have a reduced time window over which we can monitor this reaction for this site, we are able to have fewer data points, noisier spectra, and even though in this example we're able to now get a rate constant which is consistent with the one from the other site, our errors are significantly higher, and that's just associated with having fewer data points. So I think this shows the importance of having as long a lived signals as you possibly can, because it allows you a larger time window over which you can monitor your reaction. And the last thing I should say, which is really important in this example, is the presence of the metal can really influence your kinetic binding, kinetic plots. And that's because in this example, pyruvate can bind to the metal, and you would have to account for that in your kinetic modeling. You would have to either know or fit the rate of pyruvate binding to the metal and the relaxation time of the pyruvate bound to the metal. In this case, it's much easier just to deactivate your metal catalyst with phenanthrene so that you know in this reaction time window, you're not having any pyruvate going and binding back to your metal. So we just add phenanthrene when at the same time we add the hydrogen peroxide. So just to confirm that the kinetic rate constant that we're getting from these SABRE hyperpolarized measurements is real, we also monitor this reaction by thermal NMR, which you can do using proton signals for these CH3 groups. I don't have much time, so I won't go into this in any great detail, but I just want to say that your kinetic fitting is much simpler here because you don't have to account for relaxation or radio frequency excitation, and the rate constant that we get is consistent with that from SABRE data. And also you can monitor the reaction using ultraviolet spectroscopy, and again, the rate constants that we get from these three techniques, so ultraviolet spectroscopy, thermal proton NMR and SABRE hyperpolarized carbon-13 NMR are all consistent, which is really good. We chose deliberately to monitor a reaction that was slow enough that you could follow it using alternative spectroscopic techniques. In the future, we hope to apply it to reactions that are too fast to monitor using some of these other techniques. But I think I can demonstrate the real power of SABRE perhaps in this slide, which is the signal gains that SABRE provides are sufficient to allow us to detect this short-lived reaction intermediate in just a single scan. As you can see here, signals when we do this at 260 Kelvin for this short-lived intermediate can be seen here and here in just a single scan. If you want to detect those signals using Boltzmann-derived or sort of Boltzmann-controlled NMR, then you have to do significant signal averaging, which takes time, and that means that to detect these intermediates using thermal NMR, you have to go to much lower temperatures. So I think I have a few more minutes. So I also just want to mention briefly, we're also looking at other ways in which we can expand the scope of SABRE hyperpolarization, and one way in which we can do that is using relayed proton exchange effects, which have been touched on before. So we use sort of an amine-based catalyst. Our amine binds to our iridium. We can hyperpolarize our amine sort of using SABRE, as you might expect. But then in a second step, labile NH protons on your amine can exchange with labile OH protons on an alcohol, for instance. So your alcohol can become polarized even though it isn't binding or interacting directly with your SABRE catalyst. And here is just an example of some of the signal gains we can get for propanol when we use each of these different amines as hyperpolarization carriers. So we've now applied SABRE-Relay also to a much wider range of molecules, including alcohol, sugars, and silanols, which you can read about in the references at the bottom of the slide. But really, again, I just want to focus on applications of this. We can use SABRE-Relay to hyperpolarize mixtures of alcohols. We can detect low concentrations of alcohols in just a single scan. If you want to achieve spectra with the same signal to noise or similar signal to noise using Boltzmann-controlled NMR, you have to do many, many signal averages, which takes significant time. And excitingly, the intensity of these hyperpolarized alcohol signals does increase as you increase the amount of alcohol. And we're trying to use this to determine quantitatively the amount of alcohol concentration you have in a mixture. But at the moment, that's extremely challenging to do due to the presence of competing water. Water also has exchangeable protons. It can interfere with this relayed polarization effect. So it's something that we really need to sort of bear in mind and be careful of. So I hope I have demonstrated to you that we can really expand the types of molecules that we can hyperpolarize using SABRE. We can do this using new sulfoxide-based polarization transfer catalysts for O-donor ligands. Pyruvate is just one example. We can now get just over 2,000-fold carbon-13 enhancements for pyruvate, which are much lower than you can achieve using other hyperpolarization techniques such as DNP or FLIPSAR, but we hope that these numbers can really be increased in the future by further optimization of our catalyst. I hope that we've sort of convinced people that you can hyperpolarize in principle any molecule using SABRE. It's just a matter of finding the right catalyst to do the job. We've also extended the substrate scope of SABRE using relayed proton exchange effects. And I hope I've convinced you that we can use SABRE to extract kinetic rate constants for reactions, detect short-lived intermediates, and a range of other things. So I should acknowledge all the people that helped me over the last few years at the University of York, in particular Simon Dockett, Pete Rayner, Wissam Ili here, Anaran Kennelly, Olga Semenova, Jennifer Lewis, who is here, and also Adrian Whitwood from the chemistry department for doing all our X-ray crystal structures. Our funders are listed at the bottom of the slide. For me, it's the EPSRC. Thank you very much for the opportunity to speak, for your attention, and if you have any questions, then I'll be happy to try and answer.
Thank you very much, Dr. Benjamin Tickner. Please recall that if you have any questions, please send them to me. I will start with the first question. What evidence supports the idea that there is a different catalytic species for H2 exchange versus substrate polarization in a way that's different from normal SABRE?
So, okay, it was a good question. If I go back to that slide, the polarization transfer catalyst with the pyruvate ligated to it, this one here, when we try and probe the exchange processes of the pyruvate and the hydrogen using exchange spectroscopy, we don't observe any hydrogen exchange or pyruvate exchange on the NMR timescale, which you'd be able to measure those rates. You'd see that exchange for sort of typical SABRE catalyst with pyridine. What we do know, and then we was thinking, well, how is it that we're able to see hyperpolarized pyruvate if this catalyst doesn't seem to exchange pyruvate or hydrogen on the timescale of our measurements? And the reason why we think that it's species like this bis-DMSO chloride complex, which is so important, is these are always present at equilibrium, and we observe very rapid rates of hydrogen exchange using these complexes. So we expect that they are going to play an important role in this process. So I hope that helps to answer the question.
Yes. Any further questions? I fear I am trained more in physics, and if I had a question, it would be very elementary. If not, then let's thank Dr. Benjamin Tickner.